So satellite viruses and satellite RNAs, these are two, I guess, really good supporters of the simplified cell hypothesis, mostly due to the fact that they cannot autonomously replicate. They can infect cells, but they can't hijack all of that cellular machinery to make copies of themselves. They need to be infected, that cell needs to be infected by a secondary virus in order for that satellite viruses or satellite RNAs in this context to be replicated. And so I guess example of this would be hepatitis B and hepatitis D. Hepatitis D can infect liver cells, won't be able to replicate itself within that cell until it's infected with hepatitis B. And this can actually cause a much more extreme case clinically than just hepatitis B alone, because yeah, you have more of that cell's resources being exhausted and more, I guess, budding action or lysis, depending on the context, taking place. So, cool. Delete that. All right, so what is, I guess, a generalized mechanism of infection? Well, it does vary depending on the virus that you're talking about, but let's just kind of go through a general overview of this. So viral glycoproteins are going to bind with proteins on the membrane, causing a conformational change. So just remember that the viral glycoproteins, again, this determines the host range. Host range. So let's just draw this over here. We have a viral envelope here and some glycoproteins to go with it. That's not too bad drawing, I guess. And inside of it, like all viruses, there's a capsid and a genome of some kind. We're not really playing too specific here. And then here's its host that it's going to infect, and it has receptors specifically designed, or the virus has receptors, proteins specifically designed for these receptors on the host cell plasma membrane. All right, so this binding here is going to cause a conformational change, and usually this binding here happens, one of two things will usually take place. It'll be either fuse with the actual membrane itself, or it'll be transported in through receptor-mediated endocytosis. And then once that happens, something that you're going to see almost and always, I'll just go ahead and highlight all the things that always seem to happen, is that the capsid, the protein coat containing that genome, is going to be degraded, either by some enzymes that'll go in and attack it, or it'll just come apart on its own. Now, usually if it's a DNA virus, it's going to have to hijack kinesin motor proteins in order to get to the nucleus, because that's usually where we want to go. Not all viruses need to get their genomes to the nucleus, and that's a huge advantage, I guess, in terms of getting more replications faster. And so that's one of the reasons why a lot of viruses are RNA, also the fact that they have a high mutation rate. But let's just kind of focus on this for the most part. This mechanism here blows my mind. They can grab a hold of those kinesin motor proteins and walk along those microtubules, and even if they get reach a blocking point, they can switch directions and move, making sure that they get to the destination to which they want to go. For something that's not living, that's pretty, nature's scary. Anyways, the viral production, once it gets through there and does, its thing, reproduces its genome and starts making copies of itself, will usually kill the cell either by budding and, exhausting all of its resources, or it may just end in lysis. We talked about the lytic cycle there. Cool. And this is just a general overview of what's happening here. We see that this is for influenza virus, and it's binding here. It's being receptor-mediated endocytosis, which is going to take it over to the nucleus, where it's going to be, in this context, the viral RNA will be replicated, and then the synthesis of viral proteins, which ultimately ends in the budding action here. So it's not a quick death, it's a very slow and painful death for those cells. So anyways, I do want to talk about influenza just because of historically speaking, it is an extremely important virus. It's killed more that virus alone has killed more people than any other, really probably than any other microbe in the past, I'm going to say 100 years. And the reason that it can do this is that its genome consists of 8 separate strands of single-stranded RNA, negative sense, that code for the viral proteins, which leads to a lot of the nomenclature. So let's just say that this is a virus here, and that's the capsid there. It's usually going to be an H, whatever, some type of a number, and usually an N, whatever type of number. And these are the glycoproteins for it. So why do you need a flu shot every year? Well, because they can swap their RNA. In addition, with lots of high mutation rates, they can change up their genome. So if their genome keeps changing all the time, then these glycoprotein structure will keep changing over time, and your antibodies won't be able to bind and to either signal it to be either eaten up by macrophages or just I guess made tranquil in certain contexts there. So that's why you need to get a flu shot every year, is because the influenza virus keeps mixing up and getting a lot of variation. And as you know, variation is really good for your differential reproductive success. Okay, so strains have been known to infect birds, pigs, bats, and humans. They're all mammals and, well, mammalian derivatives at least. Since the virus can mix and match its genome, every couple of years there's a really new strain can be created with pretty novel functions and a unique set of host strains. So take, for example, swine flu. The reason why the CDC was freaking out about swine flu, even though I only think 3 people died from it, was because it was a new strain. Not just a mutated, altered strain, it was a completely new strain. And so little to no immunity was in humans. The only type of people who were actually immune to swine flu were really, really elderly people who had been exposed to the 1918, 1917, whatever you would like to call it, influenza epidemic from way, way, way back then. And so they actually had some immunity because it bared a resemblance to that. But the point that I'm trying to make is that viruses can get variation in their population, and that's really, really good if you're put under any type of a selective pressure such as a human immune system. Okay. So with all this being said, you'd think that we wouldn't be able to have evolved to this point because viruses are really, really good at infecting us and making us sick. But we're not helpless. Eukaryotic cells aren't helpless, especially aside from the fact that we have a skin barrier, mucosal membranes. I'm not going to go into too much on the anatomy and physiology of how you have a defense against that, so much as I am going to go into the fact that the cells themselves have defenses against that. So yeah, we have our host immune system, cytotoxic T cells, natural killer cells, macrophages, B, T cells, whatever. But this thing here is pretty interesting. If you remember the proteasome and ubiquitin complexes, these are basically just little, I'm just going to draw them like this. I'm sure that they exist as something much different. But let's just say that I get a virus infects me and it's just, going around inside my cell trying to do whatever, and I have, say, an antibody they're attached to it, or some type of a ubiquitin complex. I'll do the ubiquitin in blue. Can help bind to that. Well, that's going to trigger that proteasome, which, depending on how much general biology you know that proteasomes destroy proteins, or they, I'm just going to say that they degrade proteins. If something is broke and it's not useful, well, we can strip down the parts for it, and that's what it's going to do. So it'll come in and it will destroy this virus and degrade it and prevent it from doing any damage within the cell, which is I found it to be quite interesting. Another thing that they have is Interferon. What a wonderful name there. So if I'm a cell and I get infected, I can secrete an interferon protein to all the neighboring cells that haven't quite been infected yet. And what this is going to do is this is going to produce large amounts of proteins that interfere with viral protein manufacturing or with the genome replication. So another function that they can do is to increase the gene expression of MHCs. MHCs, and they also increase that wonderful proteasome activity. So let's just say that I'm a cell here and I get infected with a virus, and I'm trying to stick out a flag saying, I'm sick. I'm sick. That is basically what MHC proteins do. So let's just, I'll draw in white that there. Major histocompatibility complex. They send a tag out there saying, I'm sick, cytotoxic T cells or natural killer cells, come kill me, please. And that way, this cell will die, but all of his neighboring cells won't be affected by that. They'll be alive, and they'll be able to keep doing their jobs. These are really bad drawings, I know, but just keep that in mind. So the MHC and proteasome activity increases not only odds of being identified and or destroyed by the host immune system, but certain types of interferons, such as interferon gamma, which is pretty unique, can actually activate an immune response all on their own. They don't need helper T cells to come in there and start getting things going. They can send out all kinds of cytokines and things like that that need to get the job done. So it's a very rapid, very fast response by our cells. Last thing that I want to talk about is a protein called PKR. And what this stands for is protein kinase. I wonder what kind of an enzyme that would be. RNA activated. That's not the cleverest of names. But anyways, interferons are going to start inducing their gene expression, and they're usually going to be activated by double-stranded RNA. Cytokines, certain growth factors, stress signals, certain viruses actually can produce decoys of this. But what these do is they attack this double-stranded RNA. rendering it unfunctional. But the thing is that viruses can deploy decoy versions of this so that keep these guys preoccupied while the actual double-stranded RNA of the virus can go in and do its job. So it's not a perfect process here. But it does inhibit translation. So even if, let's just say this is like a secondary thing here, even if it fails at stopping that double-stranded RNA, it can inhibit translations, which means all proteins within the cell will stop being translated. And so the cell will essentially be destroyed in the process, but again, that's good for the neighboring cells because they're not going to get infected. However, just like anything, anytime you put a selective pressure on something, viruses can make proteins that will make PKR ineffective, not only at one point, but at another point. So it's a constant back and forth battle between the two.